were in your third year, correct? All of us here have already experimented with bacteriology. All right. What were the give me a rundown of what your experience was when you were when you were doing it in your third year college days. What were the agars? Okay. And did you take it seriously? Uh-huh. Okay. What else, Cesar? All right. All right. Can you describe how you make cup cup? Is it made of chocolate? Yeah, is it really made of chocolate? Okay, it's made of blood. Okay, just making sure. All right. It's yeah, okay, just I'm just making sure. Oh, okay. That's okay. Don't worry. I'm just asking. I'm just confirming. All right. What about media that is liquid? Have you made media that is liquid, or have you had the chance to create a culture media that's liquid? What about the girls? Ah, yan. Yes. Thank you so much for the broth. Yes. So yeah, I was asking the girls, but thank you, Cesar. Okay. Thank you. What do you mean, Adrian? You were not able to create something that is liquid based, Adrian? Oh, really? Oh my gosh. That's oh, okay. So this is going to be an important um this is going to be an important topic for you, of course, because we're going to talk about the culture media. All right. So differentiate a lithotrope from from a heterotrope. So basically, a lithotrope is something that lives in the inorganic. It lives off of inorganic substances, so it could be metals or anything. And a heterotroph lives on lives off on organic substances. So there are terms that refer to them. The two terms refer to a carbon-based nutrient source for bacteria. So heterotroph would most likely live off of carbon from of carbon, whilst lithotrope, from the name itself, it lives off of rocks, the minerals from the rocks. Okay, lith, hence the term rocks. All right. So the element that is required by RNA by bacteria for RNA, protein, and DNA metabolism is, of course, the most important one. This is your nitrogen. So please don't forget nitrogen is important in bacterial physiology, primarily because it's involved in protein, RNA, and DNA metabolism. Okay? So minerals required by bacteria, of course, you have sulfur and magnesium. Please don't forget that I mentioned earlier magnesium because it's also a component. It is one of the sorry, medyo busog. Sorry. Um, Magnesium is one of the heavy metals that is transported freely in bacterial cell walls. All right. Remember earlier I mentioned it. It was an or it was this is a part. This is the a cell wall. Bacterial cell wall um, is capable of transporting magnesium on on a regular basis inside the cell. Sulfur on maybe sulfide inducing sulfide metabolizing organisms, and we were going to talk about them when we look at their TSI when we look at the TSI reactions of each organism. All right. Now, what is the term used for a salt requiring organism? Why did I ask this? A salt requiring organism or salt requiring bacteria. This is known as a halophile. A halophile. Vibrio and Staphylococcus species are considered to be halophilic organism. Hence, they can grow in mannitol. Hence, Staphylococcus can grow on mannitol salt agar, while the culture media for Vibrio species require a bit more, a bit more saltier, a saltier culture media. Okay, we'll talk about it one by one later on. Which species of Vibrio is not considered a halophile? Well, there's only two: Vibrio Vibrio cholerae and Vibrio mimicus. Yeah, Vibrio cholerae and Vibrio mimicus. Now, Mycobacterium tuberculosis is an organism with an increased concentration that requires an increased concentration of protein. It requires an increased concentration of protein. The growth requirement is the reason why technologists use this as a component of a culture of the culture media. This requirement. Most culture media for traditional culture media for the growth of microbacterium tuberculosis is actually based off an egg, whole egg. If you look at the literature, a lot of them requires an egg because of an increased requirement for protein, right? Specifically albumin. Now, in order to cultivate another growth requirement, in order for this now for this specific organisms, Haemophilus, it requires the XNV factor. XNV factor. Another name for XNV factor is your hemine for factor X, hemine or porphyrin, and factor B is NAD, nicotinamine adenine dinucleotide. All right. Please don't forget the growth requirements, factor X and factor V, just the other terms. And also don't forget the other terms, hemine or porphyrin, factor V is NAD. Hence, the reason why we can we can only grow Haemophilus organism in what routine culture media. I will give you guys I will give you guys choices. The growth of Haemophilus influenzae requires this particular can be seen in the growth of Haemophilus is supplemented or enhanced in which routine culture media. Makonki, uh, this one, and what's the other one? Manitol. Okay? So, these are your choices. Oy, galit na galit si Cesar. Cap, sir, cap! Ganon. Medyo naka... Cesar medyo galit. So these are your routine culture media. Hindi naman, hindi naman. All right. Medyo kasi naka-headset kasi ako, Cesar. Medyo na ano ako, medyo nabigi ako. Ah, nasa clinic pala si Cesar. All right. Mamaya kita interviewin. So all right, all right. So a lot of people answered cap. Yes, that is correct. Why? Because species of Haemophilus requires the growth of factor X, which can only be released when you lyse the cells in your sheep blood agar. And in order and a lysed sheep blood agar will result to discoloration of the blood. The blood will be discolored and it will be colored brown. The brown color is the reason why we call it sheep agar. Chocolate agar. All right? That's the reason why we call it chocolate agar. And with the lysed cell, they will release hemine or porphyrins in the media. All right? Hindi tayo naglalagay ng sneakers, hindi tayo naglalagay ng ano pa bang mga pwedeng ilagay? Sneakers, hindi din naglalagay ng Hershey's or hindi din naglalagay ng Toblerone. Although I think bacteria would enjoy that much, but no, we don't put it in there. All right? Because I remember one of my professors were trying to check if we were listening to her discussion. And one of the questions was stated this way. True or false? It's a true or false question. Chocolate agar plates require the addition of 90% chocolate, specifically Hershey's. She put Hershey's. I found it funny back then, but none of my some of my classmates found it funny, but those who weren't listening, apparently they answered true. So yeah, a lot of professors will try to put random questions like these in their exams just to check if their students are listening. And please, baka mamaya utuin kayo ni Sir Marco, maglagay ng ganon, di ba? All right. So what are the sources of factor V? Factor V is also known as hemine. All right. NAD, sorry. Factor V is NAD, nicotinamine adenine dinucleotide. All right. Uh, what are the sources? So we have plant sources also, yeah, and other bacteria can produce factor V. Um, we'll talk about why factor V can be produced by why is important to why it's important that I mentioned some bacteria produce factor B, but another source would be potatoes and yeast. So during the manufacturing stages of your culture medias, they add this just for the sake of growing, just for the sake of isolating factor V. Okay, so some bacteria produce factor V. Examples of bacteria that produce factor V is what you call there's three organisms: Staphylococcus, Neisseria, and pneumococcus or pneumococci. There's a lot of pneumococcus, so we're gonna put this under scrutiny for a while, but I'll give you guys an example. Moraxella cataralis is one. Branhamella cataralis is the other, another term for this one. So just an example of a pneumococcus. All right. So what would be the appearance of an organism that's grown with near factor V producing organism? This is actually known as satelliting test. I'll give you guys an example of what it looks like in the culture media because I don't have a plate with me. I should have brought one early when I took a day off yesterday. Um, so I'll use a pen later. All right, so this, for example, is your culture media. Imagine this is your Haemophilus organism rather. So a small part of them, usually they are fastidious organism, which means they require a lot of tender loving care. All right. So when I say something is fastidious or is a fastidious organism, they require a lot of care to grow them. So they require CO2, and then they can only grow on cap, and then and other requirements such as factor V. All right. So now that we have an initial inoculum, we're going to streak uh, we're going to streak a known control of we're going to streak a known control of Staphylococcus, which is colored green. All right, right here.